Hi, everybody. So this is lecture 7.1. This should be for week 7 and for our module 7. In this particular lecture, I'm going to be talking about how we diagnose viruses. So you might be curious, especially since probably most of us at this point have tried a rapid SARS-CoV-2 test, or maybe you or somebody you know has had a quick strep test. So we're going to go through that. I will bring up the PowerPoint and also do the whiteboard today. As I said, today we're going to be talking about the diagnosis of viral infections. Some of the learning objectives for this are to know the 5 approaches that are used to diagnose viral infections, and we'll talk about kind of the strengths and weaknesses of each of those. We'll also talk about a type of test called an ELISA, and I will explain what the ELISA stands for. And we'll talk about that there being a difference between doing a direct or an indirect ELISA. And then the last thing I really want you to think about as we're going through this is that you should, at the end of this, be able to explain what part of our adaptive immune response both the antigen and antibody detection is based on. So I want you to think about what we talked about in week 6 and kind of talking about that specificity that we see within the adaptive immunity. So with our first slide, we're going to talk about how viral diagnostics are done in a clinical laboratory. So for example, if you were to go in and see a physician. So greater than 60% of all infectious diseases that physicians see are caused by viruses. Now, most commonly, they're caused by things like the rhinovirus, and it's just a cold. But what's really important is if we're looking at something beyond the common cold, we want accurate and rapid or fast detection so that we can diagnose what it is and hopefully do a successful treatment. So probably many of you know that with things like influenza or with SARS-CoV-2, we do have treatments, but often they have to be taken very early on in the infection. So that's why it's important that not only is it accurate, but that it would be done quickly. Some factors that could influence the laboratory outcome are the type and quality of specimen. So how good of a sample did we get, and were we able to get enough that we can test? We also want to think of how quickly we do the test on that sample. So transport condition, if it has to be transported to a lab, and the amount of time between the collection of it and the actual testing on it. So don't get overwhelmed by this. I just briefly want to kind of go over just a few of these just to give you some idea, but I won't be testing on this. But thinking about the way that we think about how we might go about testing for a potential virus. So for example, respiratory viruses, we think of often having a sore throat or bronchitis, a cough, things like that. There's many potential viruses that could cause this, and so this is a list of them. So most commonly when we go on to test this, we want to get a specimen either from their throat or we want to do it from their nasal passages. These are usually the best ways that we can collect that. There are times where we might want to collect from the lower respiratory tract, but those would tend to be ones that are really infecting that portion. Maybe some other ones, for example, like a central nervous system one that we haven't talked about, but we'll likely talk about later. So if you've heard of spinal meningitis or encephalitis, the swelling of the brain, often we have to get cerebrospinal fluid from either the spinal cord or for the brain. So this would be a much more invasive way to actually check it. But you'll see too that we have a lot that we do serum or blood for. And often these are ones that we actually want to get, that these are viruses that we can detect in the bloodstream. And so we would need to take a blood sample from somebody. So keeping in mind that each of these would be specific to the type of virus that we're looking at. With that, I want to talk about the 5 different approaches that we have. And I want to just briefly go to my whiteboard and kind of, I'm going to briefly talk about the pros and cons before we go through each of these, because I want you to think about each of these. So as we think about these, there are really kind of 5 approaches for diagnosing a viral infection, right? A viral infection. And I'll kind of briefly talk about it, but we'll get to these in just a second too in the lecture. The first one is what we call microscopy. So this means that we look at a sample under the microscope. The problem for this, for many of the viruses that we have, you have to have a very high power microscope to look at these, and you have to be trained to know what to look for. So this is difficult because viruses are very small. So we might do this for a bacterial infection because bacteria are bigger. For viruses, you really have to be kind of a specialized lab for this. The second way that we might look at this is that we would look for the viral antigen detection. And if you remember when we talked about what an antigen is, that's anything that a virus might have that the body responds to with the immune response. So basically, it's something about the virus that looks foreign and our body wants to respond to it. So when we think of a viral antigen detection, we're looking for something likely a spike protein or a protein on the nucleocapsid that would let us know that that virus is present. This is actually what we look for when we are looking at rapid antigen tests. So if you've done the COVID-19 at home where you swab your nose and you put it in, it's actually looking to see if those spikes or that nucleocapsid proteins are there. And this is also something that we would see in what is called a direct ELISA test. And we'll get into that in a little bit more detail in a second. So those are our first two. And this one, the viral antigen detection, is one of the better ways that we have to detect viruses. I will say that it's not always the most specific, or not specific, but the most the best at picking up small amounts. So if you've just been exposed and you don't have very much viruses, it doesn't work as well as some of the other ones. And so we'll talk about some of the other ones. The third one is that we might do what is called a culture of the virus. And basically what that means is that you take the sample from the person and you put it on top of cells. And then we wait like 3 to 5 days to see if the cells are destroyed by a viral infection. This one's not so great because this one takes time to grow cells, right? And see if they are damaged, if a virus damages them. So when I did my research on HIV, I did cell culturing, and we did this style of doing it, and it allowed us to see when we had medications present, if the culture survived because the medication was present, or do we have lots of those cells dying because of viral infection. So really, it's more of one of those ones we'd see in a lab rather than in a clinical lab. So the fourth way that we might do this is that we might do what is called nucleic acid detection. And nucleic acids are things like DNA and RNA. So in this case, this is when we would be looking for. So it looks for the viral genome. Now, the really cool thing about this, and I'll have a video up this week along with some videos on ELISA and rapid antigen testing so that you can see kind of how these work. The really cool thing about this is that you don't need very much virus material to actually recognize that it's there. So if you went in and you had what is called a PCR or RT-PCR test, and I'll put these, so PCR, RT-PCR, or even if you had what was called an NAAT test, this is actually what they were doing when they were looking for the coronavirus. Now, usually it takes a little bit longer than a rapid antigen test, but it's much better at picking up very low amounts of that virus. So this is kind of what we would call the gold standard. It's the one that if you get a positive test with this, you can be assured that you have whatever that virus is. So we'll talk about that as well, and there will be videos up in this module. The last one that we can do is we can actually look for antibody detection. Now, antibodies, if you remember from last week, are made by us. In response to an infection, our body will produce antibodies. So this is really what we would call an indirect method, meaning that we're not testing to see if you have the virus. We're testing to see if your body has the antibody to the virus. And the problem with this is that the virus may be gone. And your body is still making antibodies because it was still fighting it off, but you may not be infectious. But this is a good way to see if somebody has been exposed to something. So if you've ever had a TB test, often what we do is see if you have antibodies to tuberculosis, and that means that either you were exposed to it at one time or you may have a current infection, but we don't really have a way of knowing without doing further testing. So these are the 5 methods, and I'm going to kind of go through each of these on the PowerPoint as well. So we'll go back to our PowerPoint and we'll walk through each of these. So the first one I talked about was microscopy. We can use a light microscope. In this case, we can actually even look at intracellular inclusion, so inside of the cell. We can also look at something called immunohistochemistry, which means we can look at what is being shown on the outside of the host cells to the immune system. Again, this wouldn't be something you'd see typically in most clinical offices because most people aren't trained on these. We can also have an electron microscopy, and this allows us to actually see a virus or a virion on its own. These are very powerful microscopes, very expensive, which means they're not commonly found in every doctor's office. You can also have immunoelectron microscopy. So if you've seen all those great pictures that are zoomed in on like a virus particle of SARS-CoV-2 or HIV, these were done with electron microscopes, and we don't have that really clinically. So that's why I was saying this is of limited value in a doctor's office, but maybe in a research institute, this would be something we'd use quite a bit. We also have what we call ELISA, and ELISA stands for Enzyme-Linked Immunoabsorbent Assay. It's a lot, so that's why we shorten it to ELISA. But basically what you want to think about with the ELISA, and this one would have been kind of our second of those rapid tests. With an ELISA, we're either doing one of 2 things. On one side, we are doing what I would call a indirect method, and on this side, we would be doing a direct ELISA. So let's say I suspect that you have some sort of an infection. I may take a sample from you, assuming that the antigens, the spike proteins or whatever it is, are in it. And then I put it into a container that has antibodies that are specific for that particular virus. If you have that virus, what we would expect is that your little virus particles would attach to it. And then we can add another addition, which is an enzyme that will bind to the virus as well. And this one has this kind of chemical flag on it that when we add another solution, it changes the color of this. So if we see a change in color, that's a positive test. It tells us the person had the antigens. Now, if I had added your sample up here and there was no virus there, nothing would have bound here. These enzyme labeled antibodies wouldn't have bound, we would have washed it out, and we added the substrate, it wouldn't change the color, and that would be a negative test. But in this case on the right, this is a direct assay, which means that we are looking for the antigen. We can also do what's called an indirect. Remember, we can look for antibodies, which tells us that the person either has a current or past infection. In this case, we'd take samples of the person and we would put, this would be of blood, and we would put it in with the actual virus antigen. If it binds to that, it means it's specific for that particular virus. And then again, we add this enzyme labeled antibody in here that when we add a substrate will turn a color again if it's positive. If you didn't have the antibodies, none of the other things would add in, and when we wash it out, there wouldn't be a reaction and it would be colorless. The nice thing about the ELISA is that it's relatively inexpensive because this actually takes place in these very small tubes or microtiter plates, so it doesn't take very much chemicals. A lab tech can do this. It's not something that's super complicated, and we get a very fast turnaround, usually within hours, minutes to hours rather than days. So this is a great way to do it as well. I do have a video up because I think sometimes I will have a video, for both an indirect and a direct ELISA, because I think often with animation, it makes it easier to understand. And before I go past that, I do want to say that the rapid test that you do also for something like SARS-CoV-2 is looking for those antigens. I do have a video of that up showing you how those actually work. So again, it is that antibody to antigen reaction, the idea that these will bind to each other, and then we can add something that will give us a color to show us that it was a positive test. The third way that we can culture or look for viruses is we can do something called a cell culture. Remember, viruses don't replicate unless they're inside of cells. So one of the ways we can do this is we can actually grow cells in order to then grow viruses. So cell cultures are used to isolate viruses and identify them. And what we look for is basically the destruction of cells. So if you have maybe a plate that has all these cells lining the bottom of it, and then we add virus to it, the viruses, when they infect the cells, will burst the cells and kill off the cells. And we end up getting these spaces of empty parts on our plates because there are no longer cells there. And that's what is the cytopathic effect. So we look for the virus to destroy the cells. They do have a faster version of this where we can actually centrifuge the virus so it lands on those cells much more quickly and infects them more quickly. And this will allow us to actually look for those viral antigens. Again, this is something that's a little bit technical, and you would have to have the facilities to grow cell cultures in order to grow the virus. As I said, I did this in my graduate work, but this was set up in a diagnostic lab or in kind of a scientific lab, not really a clinical setting. This is giving you some idea of what that would look like then, right? We take a specimen from the host, we drop it in on top of these cells that are at the bottom of the liquid. We centrifuge it so that it basically gets those viruses to drop onto the cells fast. But remember, this can take about 1 to 5 days waiting for the virus to basically go through the replication cycle and burst out and kill the cells. What we can do then is we can look at this under the microscope and count how many places are missing cells, and that tells us about how much virus is there. It's an interesting way to do it, but probably not practical in a clinical setting. Now, when we talk about nucleic acid detection, as I said, this is probably ways that you guys may have been tested if you needed to go to a pharmacy or you had to travel to get your SARS-CoV-2 test result. And so basically what we do here is we take a sample from the host, so from your nose maybe, and we basically do amplification looking for a set part of the RNA of SARS-CoV-2. We basically amplify it, make many, many copies of it so that then we can detect if it's present. So this allows us to detect the actual genome of a given virus. And it's very specific because we only look for that section of the DNA or RNA of the virus. It wouldn't have a positive test on our cells or on another virus. The ways that are most commonly done are PCR and RT-PCR. I have a video up in this module as well that shows this in an animation, so hopefully it helps you understand. It's a great way to diagnose because, as I said, this is the gold standard. We can have a very small sample and still get a result because it's so good at being able to detect that. It's also, besides diagnosis, used to manage patients. So we use PCR to look at HIV or hepatitis C patients to detect how much of the virus is circulating in their body and to monitor how high the viral load is. So if we get a very strong PCR test, it tells us that there's more virus than if we had a low one. The last way that I want to talk about doing it is that idea of the antibody detection. We talked about the fact that we can do that with ELISA, but we have other ways that we can do that as well. So we can look for the presence of antibodies as an indirect method. Remember, this isn't going to tell us if you currently have the viral infection because we don't know if those antibodies are because you are currently infected or maybe you've already cleared the infection and those antibodies are still around. We can look at the type of antibodies present. So if it's a recent viral infection, we would expect it to be an IgM antibody. And if it's a reinfection with the same virus or we're further into the infection, we would expect it to be IgG. I don't expect you to memorize that. Just know that we can detect, though, antibodies in a person as well. So the methods that we do this, as I said, the ELISA, the indirect ELISA method. We can also do something called an indirect immunofluorescent assay. Basically, we can look to tag the antibodies with fluorescent molecules, and then when they light up, we have machines that can measure that. We can also do what's called a Western blot, and it's a way to also look for those antibodies or proteins that would be found in a person's bloodstream. This is giving you some idea of the different ways that we do this, kind of the basic concept of each test. So I've gone over this, but this might be a good way to kind of review. And then this also has the advantages and disadvantages of each of these. As I said, antigen detection is a really nice one because it's quite quick, and usually it can tell us if we have a current infection, but it often can have some false positives, and so that can be a concern. The other one that's a really nice one is that nucleic acid detection. It does take usually hours to even a day to actually do the test. But the super nice thing about it is it's very sensitive. And that's a great one for when we're looking at SARS-CoV-2. With that, I do want to just briefly talk a little bit about the idea of how we can test to the load, how much virus is in a person's body, and also if the particular virus is responding to a drug that we would like to treat it with. So we can do either what's called a PCR strategy, and this is the way that we measure to see how much virus is in a person. And we can compare their viral load measurement before we give them the treatment, and then give them the treatment and see if we get a reduction, and that tells us if the drug is actually working. The other way we can do it is using, again, those cell cultures, where basically we can have cell cultures infected before we infect them, we put the drug that we want to treat with, and then we infect them, and we see if we can actually get a decrease or prevention of any of the cells being killed by the virus. This also gives us a good chance to see if the medication is damaging the cells. So these are some of the other ways that we can use diagnoses or diagnostic tests. And with that, I'd like to pause and give you guys a quick checking in. Feel free to pause it here, write down the answers to this, and then come back and turn it on, and I'll walk through what the answers are for this. So for the checking in, my first question is, name at least 2 specimens that can be used to diagnose viral infection. Throat swab would be one, the nasal swab would be another. We can also do a blood test is a possibility as well. The second question is, does indirect ELISA detect antigens or antibodies? This particular test would detect antibodies. The direct ELISA would detect antigens. The last question is, which tests detect nucleic acids of viruses? The one that we think of most commonly is an RT-PCR or PCR. And also sometimes you'll see it written as an NAAT, and that's just a nucleic acid assay test. Hopefully that was helpful. And please note, though, that I did put some videos up that have the tests, the diagnostic tests with the animation, so often that helps understand them better. Let me know if you have any questions. Thank you.